Hi everyone, so today we're going to go over the process of translation, but before we do that, we first need to understand tRNA. tRNA stands for transfer RNA, and from its name you kind of imply that it has something to do with the transfer of molecules, and that is obviously correct. tRNA is the molecule that brings the corresponding amino acid to our mRNA codon, and basically is the molecule that helps create a protein. Now, to look at the structure of tRNA, it's kind of this cross type thing at the end of each rectangle. So at the end of each side there are specific circles. More specifically, you want to look at the bottom circle right here. This is where the anticodon is attached. What is the anticodon? So the anticodon is basically the opposite or the complementary base sequence to our codon. We know that a codon is a specific set of 3 bases, and as an example we have GCC right here. So if that's our mRNA codon, then our anticodon will be complementary to GCC, which is going to be CGG, right? And this is the anticodon. I just write anti, but that's the anticodon, and this specific code is at the bottom of our tRNA molecule, or it's going to be attached onto a tRNA molecule. Well, why is that important? Well, it's important because it ties into the process of translation. So essentially what happens first in translation is that a ribosome attaches onto the mRNA strand that has left the nucleus, right? So let's just draw the RNA strand here, and actually let's draw that a little bit bigger. So we have our RNA molecule right here. Oh, not RNA, this is our ribosome, and we have our mRNA strand right here. So this mRNA strand contains this specific code up here, and it's going to be translated. Essentially, what first happens is that the ribosome is going to start reading the code. So it's going to start from UAG, which is one of the start codons. It's one of the start codons that code for a specific amino acid. So the ribosome is going to read UAG, and it's going to signal to bring its corresponding tRNA molecule. Well, how do we know that it's the correct tRNA molecule? Well, UAG, it kind of identifies this by seeing whether it's anticodon, or whether the tRNA molecule's anticodon matches the codon specifically on the mRNA strand. So it's going to see, does it have the matching does it have the anticodon for UAG, which the anticodon for UAG is going to be AUC, yeah? So the tRNA molecule is going to see, oh, do I have AUC? And it's either going to be yes or no. If it does have the matching codon, what it's going to do is that tRNA molecule is going to go to its corresponding codon, which is, the first one is UAG, right? So it's going to go to the corresponding codon, and then it's going to attach its anticodon to the codon. So you have UAG here, and so the corresponding anticodon is going to be AUC, right? So that's the anticodon on the tRNA molecule. And then now that it's done this, let's look at the top structure of the thing that's on the top, right? This is where the corresponding amino acid to that codon is going to be located, right? So as we know, one codon codes for an amino acid. So that specific amino acid is also located on the tRNA molecule. So the tRNA molecule is right here. It's joined its anticodon. So what it's also going to do is join its amino acid right there. So that's our first amino acid. Now the ribosome is going to move to the next codon. So we've done this codon, we can cut it off. So it's going to move to the next codon, which is GCG. Now what's going to happen is that it's going to signal to tRNA, do you have the anticodon for GCG, which is going to be CGC, right? So it's going to, we need a tRNA molecule that has CGC. So the tRNA molecule is going to be like, yes, I have it. It's going to come. It's going to attach its anticodon to things that say GCG is right there. It's going to attach its anticodon, which is CGC, and it's going to give its next amino acid. Whatever GCG codes for, it's going to attach to the next amino acid there. And what happens is that these 2 amino acids are going to form together. They're going to link together through peptide bonds. And this process happens and it starts to do this across the whole mRNA strand until it reaches a stop codon, right? So once it reaches a stop codon, it stops translating. And then at that point, you're going to have all these different amino acids linked together, formed by peptide bonds. They join together by peptide bonds, right? So then what happens is that this whole amino acid chain sequence, right, that is what we call polypeptide. A polypeptide is just multiple amino acids joined together. And that is essentially the whole process of translation, where to simply put it, ribosome attaches onto mRNA strand, it starts reading the strand, it brings its corresponding tRNA molecule to the specific strand, the tRNA molecule binds its anticodon to the actual codon, and then it releases the corresponding amino acid, and these build up, it creates these amino acids joined together through peptide bonds, and the polypeptide will fold up on itself, create more bonds, and become a functioning protein. That's essentially the whole process of translation.